The molecules known as lipids include fats, oils, and waxes. Now, the structures among the lipids can be quite variable, but the one characteristic that they all have in common is that they are nonpolar or hydrophobic. They do not mix well with water. Now, why is that? Well, most of the lipids are going to include a subunit known as a fatty acid. Here are two fatty acids over here on the right. If you look very closely at these fatty acids, you'll notice that they are mainly made up of these CH3 groups. If you remember from chapter 4, CH3 groups are called methyls, and they are extremely hydrophobic, nonpolar and hydrophobic. So because they have these fatty acids attached to them, most lipids are also very, very hydrophobic. And you guys knew that, right? Oil floats on water. It doesn't like to mix with water. We tend to think of lipids as being really bad for us, and that's because we tend to get way too many lipids in our average diets. But biologically speaking, lipids have a number of really important functions. First and foremost, they are really important energy storage molecules. Now, whereas carbohydrates can be broken down quickly and are good for short-term energy, fats are harder to break down, so they are used for long-term energy storage. That's the reason that animals such as bears tend to put on a lot of extra fat to help them survive the winter. Second, lipids make excellent waterproof coverings. So for instance, you have oils on the outside of your skin, and that helps waterproof your body. Some leaves have wax on the outside, and that helps waterproof them as well. Next, lipids are the major component of cell membranes, which we'll see in a minute. And fourth, lipids make up these molecules known as hormones. Hormones are important in cell communication. Lastly, lipids are really important as a form of insulation. I mean, think about it. Animals that live in really, really cold places like walruses, you never see a skinny walrus. They have these really, really thick layers of fat on their bodies, and that actually helps hold in heat. Fat is an excellent insulator. There are several different ways to categorize lipids, but these are the categories we're going to use and go through. Triglycerides, membrane lipids, fused ring steroids, and waxes. Triglycerides includes oils and fats. So this category includes things like bacon grease and lard and Crisco, but also vegetable oil, peanut oil, olive oil, those types of things. Structurally, triglycerides can have a lot of variation, but you're going to find that the ones we examine are going to have three characteristics in common. Here we have a triglyceride molecule. If you examine that molecule, you'll notice that it's only made of carbons, hydrogens, and oxygens. So you don't find anything strange attached to it like sulfur or nitrogen. Second, all triglycerides are going to have at least one, and more commonly three, of these long tails that we call fatty acids. There's three on this one. Finally, most of the triglycerides are not ring-shaped. There's so much variation in structure among the lipids that you can't really say they have true monomers like some of the other groups. But there are certain components that you will often see, and these components are definitely found in the triglycerides. So let's take a look at those. First of all, we have this molecule called glycerol. Glycerol is the head of a triglyceride. Notice that it is a 3-carbon chain, and that each carbon is attached to an OH, or a hydroxyl group. The other major component of a triglyceride are these fatty acid tails. And let's think about how many there would be in a triglyceride. There would be 3 fatty acid tails. So here are the fatty acid tails. Notice that on one end they do have a carboxyl group, which includes this little hydroxyl. And the rest of the tail consists of a long string of nonpolar methyl groups. This is what makes the molecule nonpolar or hydrophobic. Now, the presence of these little hydroxyls on either side of the molecule is what allows the tails to attach to the heads. Now remember, what kind of a reaction would we have to use to attach those tails to the heads? Well, of course, we would have to use a dehydration synthesis, and that's exactly what happens. In order to attach the tails to the heads, one side has to lose a hydroxyl, the other side loses a hydrogen forming water, and that leaves room in the bonds for these guys to get together. So here's the fully constructed triglyceride. Ooh, I almost forgot. The little bond that holds the tails onto the head is a special kind of covalent bond known as an ester linkage. So it's just a special name for the bonds in this group. I'd like you to take a moment and draw a very simple diagram of a triglyceride in your notes. Now, it doesn't have to be complicated. You don't have to draw in all the atoms or label every single thing, but I would like you to label the glycerol head and the 3 fatty acid tails. The emphasis here is on learning to recognize the shape of the molecule, because nothing else you see is going to look like a triglyceride. Give it a try. The terms saturated and unsaturated fats can actually be applied to any lipid that has a fatty acid tail. We most commonly hear these words used with triglycerides, so we're going to discuss them here. Saturated fats are mainly animal fats, although you do find them in some plant sources, like coconut oil is full of saturated fat. Saturated fats tend to be solids at room temperature, and in general, you're supposed to eat them in smaller amounts than you do unsaturated fats. So what makes these saturated? Well, to understand that, we need to look at the fatty acid tails. Now, this fatty acid tail is written in shorthand, as we went over in class, so nothing's labeled here. But what I want you to see is that it's a string of carbons, and the carbons are all attached to one another with single bonds. Because they are attached with single bonds, it means they can be attached to as many hydrogens as possible. So typically, each carbon will be attached to 2 or 3 hydrogens. This tail is saturated with hydrogens. Now, the presence of those single bonds gives the tails a nice straight shape. So what that means is that if you put a couple of these guys together with their nice straight tails, they pack together really closely, and that's what makes them a solid at room temperature. So saturated fatty acids have only single bonds and many, many hydrogens. Unsaturated fatty acids tend to be plant-based. So these include things like olive oil and vegetable oil and peanut oil. They're supposed to be a little bit better for you to get in your diet because they tend to clog arteries a little bit less than the saturated fats. So what makes these guys oils and what makes them unsaturated? Well, again, we need to look at the fatty acid tail. Notice that in this fatty acid tail, we have at least one double bond. That little equal sign there means a double bond. Now look what that does to the shape of the fatty acid. It turns it, and the actual technical term for this is it kinks it. Unsaturated fats have kinky tails. Now why does that matter? Well, if you have a fat that has a kinked tail like this, and you put a bunch of these guys together and they're all kinked up, well, they tend to push each other away. So you can see how kind of gnarly this guy is here. And so the molecules push each other away, which makes them a liquid at room temperature. Now, before we move on, I need you to think back to good old chapter 4 one more time. I want you to look at this fatty acid, and I want you to look at this little section right here specifically. There was a word provided to you in chapter 4 that describes the arrangement of the hydrogens around this double bond. And I want you to see if you can figure out what it is and write it down in your notes. That last your turn question leads us directly into the topic of hydrogenation. So I'm sure you've heard of hydrogenated or partially hydrogenated fats and oils, and you've probably heard that they're not that great for you to eat. They're not particularly good for you. So what are they, and how does that relate to this your turn thing? Well, hydrogenation is the process of taking an unsaturated fatty acid like this one, with its nice little double bond, and blasting it with hydrogen atoms until that bond breaks and it straightens out the tails. Now, why would you want to do that? Well, there are some benefits to making an unsaturated fatty acid act like a saturated fatty acid, and we'll discuss that on the next slide. The term that I hope you came up with, and that last your turn, is this one, cis. Cis refers to the arrangement of atoms around a double bond, and it specifically tells us that these two elements are going to be on the same side of that double bond. The opposite of that is trans, where the two elements are on opposite sides of the double bond. So what does this have to do with hydrogenation? Well, sometimes when fats are hydrogenated, they become saturated, as we discussed before. But sometimes those double bonds will break and then reform, and they will result in what we call trans fats. And trans fats have this kind of arrangement around their double bonds. So that's where trans fats come from. There is strong evidence that trans fats are bad for us, that they help raise bad cholesterol levels in our blood, and that they increase the risk of developing heart disease, diabetes, and even stroke. So that being the case, why would companies choose to use trans fats in their food products? Well, there are some benefits. When you take an unsaturated fat and you make it behave like a saturated fat, it tends to be more shelf stable. That means that a product like peanut butter can sit on the shelf a longer amount of time without going bad. It also means that you can use it inside deep fryers longer before the oil spoils, which is kind of fun to say. It also helps prevent this kind of separation that you see. If you've ever tried to make peanut butter at home by yourself by grinding up peanuts in the blender, you might notice that after you make it and let it sit for a while, that the oil will separate out of the peanut butter and make this kind of layer that then has to be mixed back into the peanut butter in order for it to taste normal. Well, peanut butter products that use hydrogenated oils and trans fats don't have that problem. Once they're combined together, they stay combined. So they make this really smooth, nice product with a really nice texture. So you do still see trans fats being used in commercial products and in foods, but they are required to label the products when they do use them. Membrane lipids are the lipids that make up our cell membranes. There are many different types of lipids that fall in this category, but the major one that we're going to talk about are these molecules called phospholipids. Structurally, phospholipids are pretty similar to triglycerides. They include a glycerol head and fatty acid tails, just like the triglycerides. But there are some important differences. Glycerol has 3 carbons, 1, 2, 3, and in the triglycerides they were attached to 3 fatty acid tails. Here though, look at what this guy is attached to if you look closely. That's a phosphate group. So that's where this category gets the name phospholipid. It's because of the phosphate group in the head. And the presence of that phosphate will change its characteristics quite a bit. Now, because one of the carbons in the glycerol is taken up with an attachment to a phosphate group, that means there's only 2 carbons left to connect to fatty acid tails. So phospholipids have a phosphate group in the head and 2 fatty acid tails. The presence of that phosphate group in the glycerol head of the phospholipid really changes how it behaves, especially when it's mixing with water. Remember that phosphate groups are very, very polar, which means that that head is now very, very hydrophilic. However, the fatty acid tails are typical fatty acid tails. They are very, very nonpolar, very hydrophobic. So poor little phospholipids are very, very conflicted molecules, especially when it comes to their relationship to water. The head of the phospholipid is very, very polar or hydrophilic. So the head loves water. The tails, on the other hand, are very nonpolar or hydrophobic. They hate water. What happens to these guys when they're in an aqueous environment, such as you find inside or around cells? Well, if you take just a few of them and you toss them into some water, they'll make an interesting little shape like this, a little sphere. And notice how they're arranged. They've put their heads together so that the heads are touching the maximum amount of water. Simultaneously, they've squeezed their little tails together so the tails are touching the minimum amount of water. Now, if you put enough of these little phospholipids together, they will form a double membrane. This is essentially what you find around the outside of a cell. Again, notice the heads are facing out on both sides, and the tails are facing in on both sides, so they're touching the minimum water. We refer to this structure as the phospholipid bilayer because there's two layers of these guys. Now keep in mind, they're not stuck together. They're just floating in that arrangement so that the heads are touching water and the tails are away from as much water as possible. Here are a couple of diagrams of the cell membrane showing the phospholipids. Now remember that these bilayers would actually surround the entire cell. It's a three-dimensional structure, not just a little slice. But when we slice it like this, we can actually see the phospholipids inside. Here, for instance, we have a phospholipid head, and its two little tails reaching down, and on the other side, we have a phospholipid head, and the two little tails reaching up. So it makes this very, very non-polar layer in the middle, and it makes the very outside of the membrane and the very inside of the membrane polar. This structure and arrangement helps the cell control the amount of water that comes in and leaves. I'd like you to take a moment and make a simple drawing of a phospholipid. Again, you don't have to draw in all the little atoms. I'm just looking for the shape. Once you've got your molecule drawn, I'd like you to label the glycerol and the fatty acids, and I would also like you to tell me which portions of the molecule are polar and which portions are nonpolar. Bring your work to class with you. Our next category of lipids are the steroids. Now, I'm betting when you hear the word steroid, you don't think about lipids, right? You probably think about muscle growth and about cheating at sports. So what are those illegal steroids and how do they relate to this topic? Well, there are many different types of steroids. They include things like cholesterol. They also include things like hormones. Now remember what hormones do. Hormones are communication molecules. They are released into the blood, they circulate around, and they tell certain organs what to do. So the illegal steroids send the message to muscles to increase the amount of protein they contain and to grow larger and stronger, and they do in response. Certain medications, such as prednisone, also include steroid components, and those can be used to reduce inflammation. So different steroids send different messages to different organs. They all have side effects because they can all attach to many different organs. A major steroid molecule that we find inside our cells is good old cholesterol. Now we think of cholesterol as being bad for us, and that's again because we tend to get way too much of it in our diets. And if you get too much, it tends to clog arteries and things like that. But even if you could eat a diet that was completely free of cholesterol, your liver would still manufacture some. So why does it do that? Well, cholesterol is actually a really important component of our cell membranes, as we will see in a later chapter. And it's also the precursor to the other steroids used by cells. Steroids are sometimes referred to as fused ring steroids, and that's because their structures do consist of these four fused carbon rings. Now remember that these structures are written in shorthand, so there would be carbons and hydrogens attached at these corners here. They're just not drawn in to make the diagram a little simpler. I told you that cholesterol is the precursor to the other steroids found in cells. So what does that mean? Well, Another major function of cholesterol is that it is used to manufacture the other steroids. So cholesterol is used to make estrogen. Cholesterol is used to make testosterone, and so on. So again, even if you had a completely cholesterol-free diet, your liver would manufacture some because it needs it to make these other chemicals. Our last category of lipids are the waxes. Waxes are highly saturated. You can see these long saturated fatty acid tails here, which means that they are extremely hydrophobic, and they also tend to be solid at room temperature. You find waxes several different places in nature. Bees, of course, use them to build honeycombs. You also find it on the outside of leaves as a waterproof covering, and of course you find it inside your ears. It's in your ears for a reason. It's not just dirty. It's there to help catch particles so that they don't work their way in to the more sensitive parts of your ears. Incidentally, there is a genetic basis for the type of earwax that you have, and there are two major categories of earwax. There's sticky yellow earwax, and there's also dry orange earwax. So if you're curious about that, do a little Googling about it. You might be able to sort of trace your genealogy by looking at your earwax. So that's a fun project.
As we mentioned in the introduction to carbohydrates, the periodic table hides some useful secrets. This column headed by helium has no great urge to make bonds with other atoms. The column to its left wants to make one bond. The second column to its left wants to make two bonds. Third column atoms want to make three bonds, and so on. Remembering this information, let's make a fatty acid molecule. This carbon atom wants to make four bonds. So let's give it a double bond with oxygen. And let's give it a single bond to an OH molecule. That's a total of 3 bonds for the carbon. It still wants another. So let's give it another carbon for its 4th bond. But now we have the need for 3 more bonds for the new carbon. Let's give it 2 hydrogens and another carbon. Now this new carbon needs 3 more bonds, so we give it 2 hydrogens and another carbon. We could keep going, making a chain of carbons, with each carbon having 2 side bonds to hydrogen, but let's end it by adding 3 hydrogen atoms, like this. This molecule is a molecule of butyric acid. It is the shortest chain fatty acid, containing only 4 carbon atoms. Here is another fatty acid. It is a molecule of stearic acid. It contains 18 carbon atoms. Notice that all the middle carbons in the chain contain bonds to 2 hydrogen atoms. This configuration contains the maximum number of hydrogen atoms possible. So this molecule is termed saturated, as in saturated fat. An alternate possibility for an 18 carbon fatty acid is this one. It is a molecule of oleic acid. And notice there is a double bond between carbons 9 and 10. So each of those carbons have only one bond available for the side hydrogen. Since there's a possibility to add hydrogens to this configuration, we say that it is unsaturated. And since there is only one carbon double bond, we say that it is monounsaturated. If there were 2 or more, we would say that it is polyunsaturated. Also notice that the carbon double bond causes a kink in the carbon chain. All naturally occurring fatty acids with a double bond have a kink in their shape at the location of the double bond. But it is possible to manufacture fatty acids that are transformed in shape to do away with that kink. These trans fats are more stable, so food manufacturers prefer them. But without the kink, they physically pack together easily and tend to clog arteries as a result. Adding hydrogen to replace carbon double bonds is called hydrogenation. Now let's build a fat molecule from these fatty acid building blocks. We begin with a molecule of glycerol, which will form the backbone of the molecule and hold the fatty acids in place. These hydrogen atoms in the glycerol then combine with OH molecules at the end of the fatty acid chains to form 3 molecules of water, while the rest of the fatty acid chains join to the now exposed oxygen atoms. 3 fatty acids joined to the glycerin form a molecule of triglycerides. And although triglycerides can become hazardous to your health if the level of them in your bloodstream gets unnaturally high, they play an important role in metabolism as energy sources and transporters of dietary fat.